Hello, welcome to this lecture on biomathematics. We will continue to discuss how we can use mathematical techniques and ideas from statistical thermodynamics to understand some of the biological system. We have been taking DNA as an example and then learning how to calculate free energy and partition function, etc., so that these things can be used to calculate something that is experimentally measurable in a lab. We briefly mentioned that if we can get free energy, the derivatives of the free energy will give you things that is measurable in a lab. For example, if we could get free energy as a function of force or pressure, we can get the end-to-end distance of the size of a DNA. So, what we basically were saying is that if you have a DNA, let us say in a solution, and then you apply a force. So, there is medium here and you apply a force F, then you ask the question, what is the distance from this end to this end if you apply a force, and this will give you. So, if you how do we get the answer? The distance from this end to this end. So, let me call this r. This r can be computed by taking the derivative of the free energy with respect to force. So, if we can calculate the free energy and we can calculate the force, we can get the average end-to-end distance. So, if you imagine you hold this end of the DNA, there will be always thermal forces on this and the end will fluctuate a bit. So, what you will be calculating the average of this fluctuation. So, average end-to-end distance. So, r here is the end-to-end distance, what I am going to mark now, this dotted, this distance will be r. So, this is your r, and this can be calculated by taking the derivative with respect to force. So, if you do an experiment by holding a DNA and applying a force, pull the DNA, and for each force there will be an end-to-end distance, and this can be calculated by taking the derivative of the free energy with respect to the force. So, today we will discuss this here on how to calculate this free energy for a single stranded DNA. So, this relation, if you have this relation, if you have a given force and the given extension, such relation can be called force extension relation. So, you can think of this, apply a force. So, what this people call force extension relation. You apply a force and calculate how much is the extension, because this is like you may plot force and the end-to-end extension of the DNA, and we said that it can look something like this. We will see how exactly it looks like today, but this relation can be calculated by taking the derivative. So, if you do this average r can be calculated as del g by del f, and we can calculate g as a, how do we calculate this? If you know g as a function of f, then we know how to take derivative, then we can calculate r. But how do we calculate this? Of course, this has to be minus k b t log z, and how do we calculate z with force? That is what we are going to discuss today. So, basically first we will discuss free energy of a single stranded DNA under force F. So, that is what we are going to discuss now. Free energy G of F for single stranded DNA. So, what are we doing? We are going to you have a single stranded DNA, and we imagine that one end of the DNA is fixed at a point, and then the DNA is free to have anywhere. Then you apply a force. Let us say we are applying this force. So, this is one end of the DNA and the other end is somewhere here, and we are pulling in this direction. So, let this force F is applied in the x direction. So, this is F E x. So, it has the value F, the magnitude is this F, and this is applied in the x direction. So, this is the force we are going to apply on DNA, and for a given value of this F, what is this distance from this point to this point? So, we want to measure actually this distance from here to here. So, this distance, this is what we are interested in, this end to end distance for a given force. So, this is r. So, now first we have to calculate the partition function, and the partition function for a single stranded DNA. When I say single stranded DNA, what does that mean? So, we discussed that there is a bending energy for DNA, but that is only for double stranded DNA. Single stranded DNA can be thought of as a flexible chain. It does not cost any energy to bend, it is very floppy, flexible. So, bending energy is nearly 0. So, we said that the bending energy is essentially, we said that bending energy E is A into sum over I 1 minus T I dot T I plus 1. For single stranded DNA, this A is very small, so that this energy can be neglected. So, for single stranded DNA bending energy, for single stranded DNA we can take it as 0. So, there is no bending energy for single stranded DNA. Now, what else it has? What else it has energy? So, what it has is basically, if you are going to pull something here, you have to do some work to pull it. So, that energy is there, the energy, the work done by pulling it. So, always work done by applying a force is force into distance. So, there is an energy force into this change in r, this r increase in r, that much work has to be done. So, this, if you take this, so look at this diagram. I said that the force is in the x direction and r is a vector in this direction. So, if the work done by pulling, work done for pulling, the energy for pulling is basically force into the distance r. So, this is basically F E x dot r. R can be thought as r x E x, r y E y, it is plus r z d z. In 3D, you can think of it as a vector having 3 components. So, f x E x. So, as we said in vectors, E x dot E x is 1, E x dot E y is 0, E x dot E z is 0. So, the answer is F r x. So, this is the work done for pulling. So, bending energy is 0, but the work done for pulling is F r x. So, this r x can be basically turn out to be, this r x will turn out to be cos theta if you take the angles properly. So, imagine for example, let's imagine that for simplicity, let's imagine that this is our x axis and this is your y axis, and imagine that the end to end distance has some particular, the DNA is in this particular direction, and this end to end distance will have some vector r, and if the angle is theta, this is r x. So, this is r vector and this is r x, and r x is basically nothing but r cos theta. So, this angle of this is cos theta. So, f r x can be written as f into cos theta into some constant. So, with all these constants absorbed, I will call this f tilde. So, let us call this f tilde cos theta. So, this will be the energy will be f tilde cos theta, because this is cos theta. So, what do we have? We have two energies. We have energy which is basically bending energy is 0, pulling energy is F tilde cos theta. So, basically what do we have is total energy E is 0 plus F tilde cos theta is actually F tilde cos theta. So, F tilde is F into distance, some distance d, or and so F tilde cos theta is basically your energy. Okay. Now, let us write down the partition function. The partition function is basically, we said that partition function z is sum over I e power minus beta e I, or this is integral all possible states e power minus beta e. So, now E is F tilde cos theta. So, this is integral all possible states. So, that means all possible tangents, where like we discussed before, e power minus beta F tilde cos theta. So, and it turns out that like we did before, this dt integral can be written as integral dt can be written as integral d cos theta integral d phi minus 1 to plus 1 0 to 2 pi. So, this is basically converting the vector in 3D. This is basically a volume element in 3D into spherical polar coordinates. So, this is spherical theta and phi are spherical polar coordinates. So, if we do this, if we can convert dt into this particular form, the integral will turn out to be like the integral we did before, and the integral will be essentially e power. So, basically d phi, this will give you 2 pi and minus 1 to 1 d cos theta e power minus so there is a f tilde cos theta. So, this is basically f tilde cos theta, and there is a by kbt. So, let me put a beta there. So, beta f tilde cos theta. So, like we did before, the answer will turn out to be 4 pi. So, the answer will turn out to be 4 pi sin hyperbolic beta f tilde by beta f tilde, like we just did yesterday, the other day in the previous class for the calculating the partition function for a double standard DNA. Single standard DNA with force will turn out to be a similar answer, and the answer is basically f beta f by beta f whole power n. So, it depends on the force you apply, and it depends on the number of monomers you have in the DNA, how many monomers you have in the DNA. N is the length, is the chain. So, what would this give? This is your partition function, and what do we need? What we need is the free energy, and if you calculate the free energy, which is minus k b t log z, and it turns out that the free energy will be, if you do this properly, n log f tilde by k b t minus log sin hyperbolic f tilde by k b t. So, it will depend on the temperature. So, this is just taking the log of that partition function that we had wrote, which basically is e power minus sine hyperbolic f tilde by kbt by sine. So, if you take beta as 1 over kbt and then you take the log of this z and take a minus sign, you will get this particular answer. Now, what do we need is, so now you look at here, this is a function of force. So, we can write n into log of f into, so this tilde, if you properly look at it, this will turn out to be b here, f b by kbt minus log sine hyperbolic f b by kbt. So, for each, so now from this, if you calculate the derivative of this with, so this is basically our g of f. So, now we have free energy as a function of f here. Now, what we need is to get the end to end distance r average, r x average, basically in the x direction we said this. So, to get this r x average, what we need is del g by del f. If you do this calculation, then find the derivative of this g with respect to f, there is an f here, there is an f here. If you do this derivative with respect to f, the answer you would be getting is basically this is equal to n b 1 over tan hyperbolic f b by kbt, tan hyperbolic f b by kbt minus kbt by f b. This is what you get. So, here is the relation between force and the end to end distance average. So, just by calculating free energy, we got a relation between the force f, the temperature and the end to end distance. So, for a flexible DNA. So, this is for a flexible polymer. Any polymer, this is true for any polymer like DNA or even a protein which has no bending energy and only energy is to pull this. So, this is the answer you would get. So, this is a measurable quantity, you can do experimentally, you can measure this and you can see few things. So, if I take this f b by kbt as, let me call this f b by kbt as x. So, I call this f b by kbt as x. So, this is kbt by f, this will be 1 over x. So, this quantity I call it x. If I do this, I can rewrite this R x average as R x average is basically 1 over tan hyperbolic x minus 1 by x. And if I expand this when x is very small, less than 1, but this would imply that f is much less than kbt by b. If you apply a very small force, f is much less than kbt by b, that is x much less than 1, this will turn out to be R x average will be approximately equal to 1 over x, sorry, x by 3 plus something of x cube for small x plus dot dot dot. Okay, so, this is there will be some quantity of the order of x cube. So, this will be x by 3 plus something. So, what does it say? For a small force, the extension and x is basically f b by kbt. So, let us write this for very small force, for small force that is when f is much smaller than kbt by b, where b is the monomer length, the size of a single subunit. It will turn out that the end to end distance goes as x. So, R x average, the end to end distance is equal to x by 3 plus some constants, plus some factors. which is small factors which can be neglected. So, the leading factor will be x by 3 would mean that for small force this will go linearly. So, the first part is linear. So, this is we wanted to plot R x versus x, which is f. So, this is basically f b by k b t. So, this as a force this will go linearly here with a slope given by b by 3 k b t. This is the slope of this. So, you will get, if you plot r versus f for small f, you will get a straight line which the slope which is b by 3 k b t. So, this is interesting. So, now what do we have for large force? For large force what do we get is, so what do we want to look? So, we have this expression which is tan hyperbolic x minus 1 by x, and for large f, when f is very large. So, for large f, so that is f much larger than k b t by f, k b t by b. It turns out that this end to end distance will go as, so okay, this is basically scale with some factors. So, let me call this n b will be equal to 1 minus 1 by f b by k b t. So, this is what you will get 1 minus. So, this would mean that R x will go as n b times, this is what you will get, which says that it goes as 1 minus 1 by f. So, R x for large force will go like this. If you plot this, you can see that this will saturate, it will for large force. For small force it is linear, and for large force it is going to saturate to a value. For f is infinity, this is 0 and this is R x is n b. So, this is going to saturate to a value n b. So, you can see that for large force this will have this particular relation. So, from this we can figure out, given a system, how do we calculate? What we discussed is basically that given a system, how do we calculate the partition function, and how do we calculate free energy, and from free energy how do we calculate something that is measurable in a lab, what is something that is measurable experimentally. So, we had this thing called force extension relation for single stranded DNA. For double stranded DNA it is a little more complicated, because you had to also take into account the bending energy. So, we would not discuss that, but it is possible to calculate some limit of it analytically, and using you can write down formulas in some limit, but the full expression has to be calculated numerically using computer. We cannot write down an expression for double stranded DNA. If you pull a double stranded DNA, how much will be if you apply a force F, how much will it get pulled, cannot be written down as a formula analytically, but an exact formula cannot be written down. You can write down some approximate formulas, but you can still calculate the partition function in a computer. So, anything that cannot be done by pen and paper, you cannot do it using computer, and people have done it, and then they could plot the force versus, if you apply a force, how much will it get pulled, relation for this. What is the use of all this? People also do experiments by pulling DNA, and from this pulling experiments, people can understand a lot of things about structure of DNA plus various DNA protein interaction. Now, you can imagine that when the protein is bound on DNA, you can pull the DNA, and then you can know that how much force is needed to pull out this protein, and thereby understanding something about the protein DNA interactions. So, you can learn about protein DNA interaction by applying force and then calculating force extension relation. So, this can be extended now to a protein DNA case, and we can learn something about protein DNA interactions, which is a little more complex. People are doing research on it. So, I would not discuss it in a class here, but it is good to understand that by learning such things, by learning how to calculate partition function, how to calculate free energy of DNA under force, and we can extend these calculations to DNA plus protein under force. So, if you pull a DNA which is bound with a protein, for example, you can imagine that a DNA is histone protein, DNA is wrapped around histone proteins. So, you can imagine that you have a histone protein. So, this is a histone octamer and the DNA is wrapped around this histone protein. Then this DNA has various shapes. So, there could be many histone proteins. So, you could imagine that there are like many histone proteins. So, this DNA is wrapped around this and wrapped around this and wrapped around this. Now, you can apply a force here and keep this end fixed and then pull this, and then applying a force and pulling it and knowing about the end to end distance by taking into account this interaction between protein and DNA also, and plotting the how much force is needed for this to be completely stretched will tell you something about the interaction between proteins and DNA. If the interaction is very weak, small force would suffice to stretch it completely. If the interaction is very strong, you need a large force to stretch it completely. So, by studying the force extension relation of a protein DNA complex, one can understand something about the protein DNA interaction, how strongly proteins interact with DNA. And this has been being used to study various kinds of proteins, and very interesting results have come out. So, I would not go into that detail here. So, it is suffice to say that such studies can be extended to other cases also and learn many useful things. But at the moment, the aim of this course is to give you a glimpse of how do we use mathematics to predict, calculate some quantities that you would be needing to use. Now, you can ask the question that okay, in general, when there is this bending stiffness also taken into account, there are few other interesting things people write down mathematically to describe. So, we said that the energy of a worm like chain model, and we wrote this yesterday, the other day, as energy. We wrote it in two ways. We wrote integral kappa del T by del S whole square, or del square R by del S square whole square kappa, and there is an integral ds. Now, we also wrote this as in the discrete form A into sum over I 1 minus T I dot T I plus 1. Now, what is this A this kappa or K is related? So, this is related to the bending stiffness, and there is a quantity related to this known as something known as persistence length, and this is an important quantity for all biopolymers. So, there is something called persistence length, and I want to discuss this and what does that mean, and how do we write down, what is this persistence length typically means. So, what persistence length physically means is that this is the length beyond which any filament will start bending. So, if you take, imagine that if you take a very small iron rod, it will be pretty much straight. If you take a very long iron rod, so like very long, so you must have seen like long iron rods which is like 10-20 meters long, for example, used for making buildings. So, if it is very long, they will start slowly bending. Similarly, DNA or any polymer for that matter, DNA, actin, etc. When they are very short, they are very straight. When they are short, they will be very almost straight, but when they are long, they will be like, there will be bending. So, if you take the short segment from here, this will be straight, but if you take this long segment, they will be slightly bent. So, if you think of tangents here, so if you draw many tangent vectors here, so if you take all this as tangent vectors, so this is tangent 1, this is tangent 2, and like this is, let me call this tangent I. So, this and this are not in the same direction. They have started, this tangent is in this direction, this tangent is in this direction. Now, if you think of a completely bent DNA, think of this conformation. So, here the tangent is in this direction, here the tangent is in this direction. If you think about some other conformation of DNA, here the tangent is in this direction, here the tangent is in this direction. So, if you think of many such conformations and you take some tangent ith tangent, this is the ith, some tangent I, and then you take some other tangent far away and find the dot product of these tangents. So, I plus r. So, if you take tangent far away and they find the product of this, if this is a completely straight line, if the filament was filament never bent, if the filament was always straight, any two tangents will be in the same direction. So, then T I dot T I plus r will be cos theta, cos of some model, so it will be some constant times cos theta, and this angle between this, they are in the same direction. So, theta is 0. So, this will be 1, cos theta will be 1. So, this will be some, if you take this as unit vectors, this will be 1. So, if T I is unit vectors, so this answer will be 1. So, this also will be 1. On the other hand, if they are bent, this will turn out to be not 1, it will be smaller than 1. So, you can write this T I dot T I plus r in the continuum fashion, and we can write, take any tangent at a distance. So, think about it as a continuous line and take this variable starting from here, you can call this S as the length along this contour from 0 to L. So, this length along this contour is called arc length. So, if you take the length along this contour, basically S is the distance from one end of the DNA to any point, and if you take some other point d s and find the dot product of this and average it. So, this is called correlation. So, this is called tangent correlation. This is same as T I dot T I plus r, and then you find the average of it. It turns out that this will decay exponentially, and this can be shown mathematically, and this coefficient that is sitting here, and this is called the persistence length. So, L p, and this coefficient here is called the persistence. So, this is like a correlation length. So, the tangent correlation length will turn out to be L p. So, the persistence length is basically the tangent correlation length for a double stranded DNA or any polymer. So, it turns out that this L p, what happens if L p is decided by basically the physical property of the material. DNA, the material property of the DNA decides what is the persistence length of the DNA. The property of the actin decides what is the persistence length for actin. So, it turns out that the persistence length for DNA, so L p is persistence length, this tell you how stiff. So, it also turn out by this turns out to be this bending stiffness divided by kBt. So, this is bending stiffness, how stiff it is to bend by kBt. So, it turns out that this LP for DNA is 50 nanometer or 150 base pair. What does that mean? DNA beyond 150 base pair. So, persistent length is the length beyond which DNA will start bending, start wobbling, start appearing wiggly. So, beyond 150 base pair DNA will start slowly bending, but up below 150 base pair DNA typically will be pretty straight. For actin, the persistent length turns out to be of the order of micrometer. So, this is about, for actin, this is approximately equal to 10 micrometer. So, up to about 10 micrometer actin will be pretty stiff, and for microtubule this is of the order of millimeter, it is like 1000 micron. So, this will be of the up to a millimeter length, this will be very stiff. So, in the cellular length scale, this microtubule will be very stiff, actin will be less stiff, DNA will be much less stiff. So, DNA is highly packed, has to be bent, is highly bent and packed inside the nucleus. The actin is pretty straight, but not as straight as microtubule is much straighter and it is very stiff. So, this is this property called persistent length. Now, what we learned? We learned about partition function, we learned about free energy, and if we know although that if you know the partition function and the free energy, we can calculate many quantities, and force extension relation is one of the quantity that we calculated. So, we learned that from partition function we can calculate probability for the DNA to have a particular shape or any filament to have a particular shape. So, now we want to understand what is the probability that the DNA will be forming a loop. So, this is some question that we can ask, because DNA loop formation is an important point important thing in biology. So, for example, you have a DNA like this, and ask the question, what is the probability that this end and this end will. So, there is some gene here, some gene here. What is the probability that the gene and gene will come together and form something like this? So, if they come together, some protein can bind here and then repress certain genes there. So, there will be. So, what is the probability that two parts of the DNA, which is L length apart, will come together to form a loop like this? So, the answer to that can be calculated from partition function. So, what is partition function? Partition function is basically, we said that Z is e power minus beta e integral over all states. All state for us is all dt's. Now, what do we want? We want to have probability to find DNA in one particular state. So, how do we calculate that? That particular state is, so let us say you have a DNA of length L and we want this to come together, so that the end to end distance is 0. So, how do we calculate that? So, we can calculate this by calculating the following quantity. So, what we said that the probability is e power minus beta e divided by z. So, we can calculate e power minus beta energy such a way that r is equal to 0 divided by z. This will give us the answer. In general, we can also calculate what is the probability to have DNA to have any end to end distance r. So, this is basically probability that DNA or protein or any polymer will have end to end distance equal to r. So, read it as for this is applicable for DNA, protein and all that. So, what is the probability that two parts of the DNA or protein will be r distance apart? Because even in the case of protein, this is very important. By calculating this, one can learn something about the shape of the protein, and shape of the protein decides the function of the protein. So, what is the probability that two ends of the protein will be r distance apart? This can be calculated by calculating the energy and then calculating the partition function and then using the formula to calculate the probability. So, it turns out that if you take DNA for example, if you take DNA of various lengths, if the DNA is very short, we said that the DNA will be pretty stiff because it is unable to bend. So, for short DNA, if you plot the end to end distance, it will turn out to be something like this. So, this is P of r versus r. The DNA will be most, this is r is equal to L, the total length, where r is the end to end distance. So, most of the time the DNA would be r will be very close to L. So, this is short DNA. For small DNA, which is length much less than the persistence length, the DNA would look like this. The end to end vector distribution would look like this. So, you see that the probability that the r is equal to 0, that means the looping probability, the probability that r is 0, these two ends come together is very small. It's not 0, but very close to 0. It's very small. Now, so this is the end to end distance for the probability to have end to end distance for a short DNA. For a very long DNA, what happens is that you can imagine that for long DNA, they will take all kinds of shape. So, very highly probable that these two ends come together. So, for long DNA, the long DNA, when the length of the DNA is much greater than the persistence length, this will look something like this. This is P of r versus r. The probability that r is 0 is pretty high, and the probability that to have very long to completely straight. So, this is r is equal to L will be very small. So, this will look like actually for very large r, this will be a Gaussian function which will be e power minus some constant alpha into r square. This will be this particular somewhere of the intermediate length, you will get something in the middle. So, for some length, so for a DNA for which length is comparable to the persistence length, if L is comparable to the persistence length, what you will get is something like this. You will get something like this. So, P of r versus r. For some intermediate length, you will get something like this. Okay, so this is not 0. Actually, the 0 will be somewhere here. So, this is some positive value, and then you will get finally reaching this 0. Okay, so you can imagine this for this intermediate length DNA. Now, you can ask the question, what is the probability that it will come together? So, this is now you can plot P of r equal to 0. This all can be calculated either numerically or using the same techniques that we discussed versus L. It turns out that it has some behavior which is non-monotonic. That means it depends, it increases and decreases. So, it increases and decreases in a particular manner. So, I would not discuss the detail here, but this has a non-monotonic shape. That means, the probability there is a particular length, and this length turns out to be close to 4 times the persistent length, which is very close to 500 base pair. This is close to 500 base pair. This has maximum probability to form loop like this. So, for very short DNA is highly less probable. For large DNA, this is again much less probable. So, this is some according to the worm like chain model of the DNA, this is the P of R. What does this mean? This means that this basically this is due to, there are 2 terms in free energy, as we said, there is an energy and an entropy. This is E minus T s or G minus H minus T s is the free energy. So, when we write free energy, so this is another way of thinking about it. When we write G, this is basically H minus T s, or F is E minus T s. So, there is entropy and energy. This is also energy and entropy. So, energy does not want the DNA to bend, because if you bend the DNA, it costs a lot of energy. So, energy for the short filaments, energy dominates. So, that is the first part of the curve. So, this is basically this part is basically where the energy dominates. So, the DNA behaves according to, like, the energy part of the free energy dominates, and here the entropy dominates. So, and somewhere in the middle, it is a combination between energy and entropy, which makes it non-monotonic. So, pretty much every system that you can think of in biology can be thought of as some combination between energy and entropy. So, energy wants it one way and entropy wants it the other way. As we have discussed in statistical thermodynamics, that always free energy will be a combination of energy and entropy, and they will compete. So, and we want a state for which the free energy is minimum. So, there are many such quantities that we can compute, and we can use mathematics and statistical thermodynamics to compute all these quantities. So, this are DNA I just took as an example. One can think of many other examples and compute various other quantities, which can be seen in some of the books that we had given as a part of this syllabus. We had prescribed a set of textbooks like physical biology of the cell or biological physics, which has in detail how do we calculate this for various quantities is being described. So, you can have a look at those books and read them there. So, this is just as an example, I discussed a couple of examples. So, just to tell you that we can calculate all these quantities using ideas from mathematics applied to biological systems and applied the laws of physics. If you apply, you will get this predict quantities, and we can predict them. So, with this I will stop today's lecture and see you for the next lecture.